Yes, we do have. Thanks, Valeria. So, the mindfulness this week is, do you get pickles on your Chick-fil-A chicken sandwich, or do you not like pickles? And then so explain what, why. So, Marcos and Ariesbeth, I think, have Friendsgiving tickets. 5 buckaroos, good slamming deal. If you need to pay using some electronic method, let me know. But yeah, we're gonna have more than just Chick-fil-A. I got a whole bunch of fun, different chips and candies and treats. I got a bunch of conchas. We could do hot chocolate too. That might be kind of nice today. Hot chocolate and conchas to start out while I go get the stuff. And then I got pumpkin pie. I got this pumpkin loaf, this pumpkin spice loaf. It looks really delicious. And some other things I can't remember. You can invite other people. Yeah. So, but we need to be careful. I mean, hopefully we sell. I have 25 sandwiches and then a plate of 100 nuggets. That's supposed to feed like another 20 or something like that. So, yeah, should have. So I don't know what we want to cap it at, ticket-wise. Some people would rather have nuggets than the sandwich, probably. So maybe we could sell 30, 40 tickets. I don't know. Okay. So, yeah, bring your friends. Yeah. Maybe have some games, have some fun stuff, do some good relaxing. Okay, right before our long break. You guys excited for the break? So, this question might be asked of you about the filtering of blood from the blood into the filtrate. So this is your blood supply right here. This is the filtrate that eventually goes into your bladder and then gets emptied as your urine. So look at that diagram carefully. Tell me how many layers, how many cellular layers does material need to go through to move from your blood into the filtrate? Okay, so if you're a molecule of water, a molecule of salt, a molecule of glucose, how many layers will you need to move through to go out from the blood into the filtrate? And then, of course, explain what your favorite, if you want pickles, if you like pickles or don't like pickles, then explain why. What's your reasoning? And, it's hard to explain your reasoning preference, but try to, okay? Just give it a shot. With recording the warm-ups in case people aren't here or something like that. Must-see TV, So my best videos. Yeah, you know what? I could post these for J-Town Update. That'd be pretty good, huh? Just me going la la. Montserrat, how many layers do you see here? Going from the blood to the filtrate. There's a green thing. Okay, so the basement membrane, that's one. And there's 2 on the sides. So how many? 3? Yes. 3 layers. And the first layer is what? the first, prove that. The first layer is going to move through is a capillary wall, and that has these things I mentioned yesterday or the day before, or one of these days. Fenestrations. You taught me the word for colander. 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 Calendario? Is that not right? So the AI translator is not good. Okay. So fenestrations is like a colander, little holes. Do you see little holes? See those big holes right there? The second layer is what? The basement membrane. And then the third layer is what? These things called podocytes. Right there. On the outer wall of that Bowman's capsule, as it says, as I always said from the beginning. Don't get it wrong. Yes, you don't see it's hidden, or it's right here. Okay, so this is a little bit bigger picture. Yeah, water, salt, they all need to go through here, but there's some big holes right here to help everything move through. Okay, why don't you go ahead and draw that real quick, a real small diagram, and then label it. Yeah, wherever. Yeah. Yeah, you can in the swarm area if you're running out of space. Other places. Don't draw the whole thing. Maybe just draw, well, that's not too hard to draw. Those 3 layers showing the blood from the left, things moving out into the filtrate. Okay. So, raise your hand if you like pickles on your Chick-fil-A sandwich. So, Jane and Bree, is that it? Is that it? So, you two like pickles then? Hold it. So you don't get pickles on the sandwich. Oh. So Bree, you're the only one who likes pickles? Oh, huh? You what? Okay. So, but you like pickles on your Chick-fil-A chicken sandwich, but nobody else does. Oh, Marcos. You don't mind it. So you're neutral. Oh, is anybody else neutral? Okay. All right. Neutral. Who will not eat that if there's pickles on there? Oh, dang. So what is it about the pickles? Can you describe? They make it too soggy. Okay, so it's just too pickly. I think they soak the meat in pickle juice beforehand, so it has that pickly flavor already in there. So the sogginess is what really bothers you. And that's probably better for us because we're getting them. It'll be sitting for a while, right, before we get them there and bring them here. So it's probably, you don't want too soggy, right? Because you're already going to have the heat and everything. Okay, that's interesting. I like come back 10 years and ask you, see if, oh, really? Oh, you guys are never going to change, huh? They haven't? Or they have? Really? Where'd you hear that? Okay, you learned about that, huh? Oh, your skeleton regenerates. Your bones every 9 years, basically. It's fresh and new because it's already always being renewed. Okay, I want to take your attention. This human tissue survey. Okay, so everybody got this paper? So what you're going to do is there are many stations around the room. So what you're going to do is find a tissue, write the name, and then write the body system, okay, that's a part of. So it's over here, there's some neurons, right? What body system is that a part of? Okay, the nervous system. Here, it's adipose, it's fat tissue. Huh. So that's your integumentary system. Okay, your integuments, which is your skins, basically. So if you need some guidance of the body system, let me know. All right? But, you're going to carefully draw what you see. At every station, there is a blow-up, image of what I'm looking for you to see. Okay, so you should see something like this. Don't draw the whole thing. Don't draw a circle and draw everything in there. Just draw a few of just so you can get the basic structure down of the tissue. Now, it should take you 5-10 minutes at each station. Okay, if you are good at taking pictures through the microscope, do that. But it's going to take some time. Now, let's go over. Before I go over the microscope, this one is kind of special. This one has red blood cells, which are really small. So what I did is I put it under oil immersion, which it improves the image. It makes it a little bit bigger. But because of that, you see how this objective is gray or white? It has 100 on it. Do not move that, okay? If you move this around and move this down, it's going to get oil all over the objective. So don't mess around with any objectives here. For the other one, sometimes I say, for this one, for this pancreas, I say 100x and 400x. So I want you to move between those two objectives. Now you know how to identify the objectives. What number does the 100x have on it? It has a 10 because the eyepiece is always 10, so it's 10 times whatever's on the objective. So, and it's colored yellow. Yellow is the 10, and then the blue, the 400x, what number is that? 40. Okay, so that's the 100 and that's the 400. There's many different ways to adjust the light on the microscopes. The easiest way to do it is with the side here. This will increase and decrease it. Also at the bottom, if you twist this, it'll open like an iris, like your eye. That can, do funny things with the angles. So you can adjust that. And there's also a little switch here. You can move this back and forth to adjust the light. So there's lots of different ways to adjust the light. Easiest way is just the intensity here. But there's down here, and there's this diaphragm there. All right. Try to adjust the eyepieces so you can see with 2 eyes. If you get them adjusted with your 2 eyes, it's going to be a lot less fatiguing than if you try to go with 1 eye. So I really want you got to spend some time in front of the microscope, really dive into it, okay? Get used to it, all right? Be comfortable. I want you to be able to switch between objectives, get that focus nice and sharp. If you want them, they're already centered and directly at the areas I want you to look, but if you want to look around a little bit, I want you to feel comfortable doing that. I can always get it back to where I need it to be if you get off where you want to go. So remember, these knobs right here will very carefully move your slide around. So if you want to kind of, if one part interests you a little bit, then I would say move it around a little bit to hone in on that. And remember, you'll mostly be using the fine adjustment, which is the smaller knob right here. On the outside is the coarse adjustment. Okay? All right? It should take you some time at each station. Okay, really get into it. Now, for example, the adipose tissue, what does it look like? Looks like a bunch of what? Circles, kind of bubbles. What does fat do? What's its job? What's its function? The structure is it's circular. It looks like just kind of, an area inside. What's the function of fat in your body? What does it do for us? It can provide warmth, insulation from temperature, but also shocks, right? If we get hit, kind of provides. That's why you have your gut, your intestines are very sensitive to getting hit. That's why we all have a lot of fat in our gut and our abdomen to protect our intestines from getting damaged and jostled. But what else do we need fat for? What does it give us? Energy, right? So the structure and function, you might say, is related because inside the cell it can store more fat inside. It'll get bigger and shrink based on how much energy it's storing. Okay, so these cells are basically for storage and to provide a layer. So these cells, they're designed well. They just increase in size to store more fat and then decrease as that fat gets used up. As far as the nerves go, you'll notice the cells, they'll be in purple. They'll have kind of little strings coming from them, right? And I talked to you about this before. What do nerves do? They send signals, right? They got to communicate. So it sends out those physical connections between one nerve and another, okay? So that structurally helps its function, right? It can communicate with the other cell because it's connected to it through that axon. So that's how you would explain the structure and function connection there. So I want you to do that for all of the tissues that you look at. All right? And there's no particular order. If you want to go around and look for one that interests you the most and start there, it's whatever you decide to do. Okay? And I'll probably, I went through and responded to the latest IA documents. It's the number 3 on background research, I think, or maybe it was 2. But I've been writing comments on those to help guide you. So if you haven't done those yet, please do those. So I give you ideas. Hopefully over break, you can start really thinking about that material-wise, and we can hopefully get some stuff set up before the big winter break. All right? Any questions? Oh, and I also gave you the study guide that you can start on. I will start doing that over the Thanksgiving break, so we'll take our Topic 11 tests pretty soon after you get back. Okay? Any questions? All right, let's go get in these microscopes. Let's go.